hear me? All right. Okay. First of all, I really appreciate the organizations of this really fantastic user meeting, and also for me to be part of it, because I've been known the EMSL as either user or just reading the papers. I think EMSL really has a world-class equipment. This is probably everyone knows. Has really appreciated not only the equipment, but more importantly, something I really appreciate for some years now is really outstanding staff members. Really, not only maintaining or developing new technology. So really, some of the state of art that really advancing the field of general chemistry and biology. So today I'm going to talk about our group work and also later on collaboration and later my talk with an EMSL about advancing spatial metallomics and metabolomics. So as you all know that a lot of work has been going on in genomics and proteomics. So, but of course, I'm preaching to the choirs, but also there's a lot of interest in developing small molecules, because in my opinion, that's really one of the major gaps in our understanding of biology in general, but plant biology in particular. So just again, we all know that these metal ions are very important. Also, many of the metabolites are very important. But more importantly, also something we have not talked a lot about is also so-called xenobiotics, like heavy metal ions, and also pesticide and herbicide, and also their function in the biological system also just as important, but we know much less about it. So in my opinion, that really to understand the whole biology, we need to know both metallomic and metabolomic together with proteomic, genomic together. So that's really give us a more comprehensive view about what's going on in the biology. But of course, it's not people do not care about understanding them. It's just much harder because for understanding genomics, we can design a probe, a nucleic acid probe that hybridizes to the target. For protein, generally speaking, we have available antibodies. But unfortunately, for these metal ions and small organic molecules, they're very large in numbers. If you look at the periodic tables, and also very subtle in structure differences to differentiate one metal ion from another, sometimes even different, same metal ion, but different oxidation states. But more importantly, their concentration vary very widely, and many of them very low quantities in that regard. So that's really pose challenges, but of course, also opportunities in there. So maybe I should try to use this one. I think you have a better. Yeah. Oh, this is better. Okay. Yeah. So that one has an echo for some reason. Anyway, so hopefully convince you that this is some areas of research, both of pose challenges, but of course, for people in the audience and us, it's also opportunities. So obviously, I was also preaching to the choir. So I'm really enjoying both yesterday and today's talk. A lot of work has been going on in, do this metabolomic, particularly, spatial ones, and the mass spectrometry really at the front of this. And because again, we heard a lot of particular isotope labeling really revolutionized our understanding of metabolomics. But on the other hand, most of these methods also we're going to hear more about later today on this using X-ray spectroscopy, microscopy, and spectroscopy to study metal ions, including Riti from SLL, Olga from Argonne National Labs. But again, these methods are having difficulty to providing real-time in situ information, particularly with the high spatial temporal resolutions. We're talking about less than microns because a lot of cells, as you know, in the micron size, but also the time domains. But also more importantly, also have difficulty. Many of them have provide three-dimensional informations. So that's really what we're interested in doing. To address this issue, then many people have been developing sensors, particular fluorescent sensor in doing that. But unfortunately, for small molecules like metal ion organic molecules, they have poor selectivity. If you look at periodic table, you probably know Roger Chin developed calcium sensors, and many other people developed a few other ones, but there are many other metal ion has not been developed because it's very difficult to basically have this so-called general methodologies. One is worse. Can you actually rapidly in the really a general way to be able to obtain the receptors or molecules can bind to these different target, small molecular target with high selectivity. That's really one of the major challenges. So for some years now, we actually been developing so-called DNA design and DNA aptamer and aptozymes. This was again pioneered by different groups who've been able to show that both RNA and DNA can be enzymes. But one thing that we and others recognize that these also can be nice receptor that can actually catalyze the same reaction, but in the present different metal ions, for example, or organic cofactors. So you can actually turn them in the reverse using the reaction or activity as a way to be able to know what metal ion is. So one other important thing about really kind of a foundation for our work is in vitro selections. 3 different groups developed this method where you start with a large DNA library, ask a simple question: out of this library, is any subset of sequences that are capable of forming a 3-dimensional structure that catalyze certain reactions in the present different cofactors. These cofactors can supply with different metal ions, different metabolites, or different molecules. The bottom line that if it does, you can using PCR to amplify, you can introduce a mutation, you can do iterative round of selection, mutation amplifications, you can actually be able to obtain specific sequences that are capable of working on someone. So this is, again, was proposed some years ago now. So now actually after some years development, we can make a very general methodology, allow us to obtain those DNA molecules and RNA molecules, not only bind to the large molecule, but also small molecule, including metal ion and metabolite, which is the subject of today's talk. So just to show you how much we had advanced to the degree that we can have a high selectivity and sensitivity, this is the paper we published in 2015. We show that we can this particular sodium that have very high selectivity among only sodium, but not lithium, potassium, other monovalent, divalent, or trivalent metal ions. And in the unpublished work, we just recently got another one that worked with the potassium very exclusively, but not with other metal ions either. But also, more importantly, early this year, we published another paper to show that we obtained DNA enzyme that are capable of recognizing one of them recognize only iron 3, but not iron 2. Another DNA enzyme that have only iron 2, but not iron 3. In this way, we can put 2 different fluorophores in this particular paper. We show that we can do simultaneous monitoring of the 2 different oxidation states of the same metal ion with the high selectivities. So again, we now got these kind of DNA science, but how can we turn into the sensors? This particular review article, we actually kind of give a historical overview about our development of turning these DNA science into either fluorescence sensor, colorimetric sensor, electrochemical sensors, and other types of sensors, including MR sensors. So they actually can, the nice thing about it is the same chemistry, you're just adding conjugate to different receptor signaling groups, either fluorophores or gold particles or different electrochemical agents. You can actually routinely turn them into a signal transduction depending on need in these cases. So this is just more relevant to DOE missions. In 2006, we worked with the people at Oak Ridge National Lab, and we were actually able to develop this portable meters. It's actually EP approved portable meter, allows us to actually do the survey of both groundwater, in this case soil, in the amount of uranium in the contaminated soil in Oak Ridge National Lab. And we show that we actually can get limit detection down to 11 ppt or 14 picomolars with a million-fold of selectivity. These are different concentrations. We got a minimal of million-fold, actually 100 million-fold selectivity over other type of metal ions. So allows actually to be able to do the in-field and test. And now we actually have an online image monitoring devices developed also for this monitoring of these heavy metal ions. And this is actually one of the TV stations in Minnesota, actually using this commercially available meter, was able to survey their lead in their drinking water in the school district. So this is one kind of translation research from the fundamentals in something that one can use either at home or in the school district. And more recent development is actually this portable meter is kind of nice, but still pretty relatively bulky, but also expensive. It's about $2,000 US. And actually more recently, we actually developed another technology working using thermometer as a way to signaling of the presence of other target. The way we did that is based on receptor binding, in this case DNA enzyme binding to the target, allowed to change the, because of heat generation, we can actually change the temperature. So based on pure temperature changes, you can actually monitor the amount of uranium present in the particular samples. In this case, just water, but also we can actually apply to other kind of sample unit in this cases. So then actually temperature changes now become actually signaling depending on the receptor what we're interested in doing. So so those actually are all being commercialized, already commercialized. So in the past 10 years or so, my group is interested in more importantly using these kind of DNAs and aptamer to understand more fundamental biology, including plant biology. But to do that, then as you know, we have to overcome some barriers to do that. First of all, I had to find a way to basically overcome delivery issues, overcome something similar to amplification issues, quantification using FRET, but also more importantly, controls, right? We want to spatial temporal controls. We actually developed something we can do optical controls, near-R photothermal controls, and other type of enzymatic control inside so that we can actually turn on, turn off at a specific location of the cell, but also timing all the decision that we're doing that. Again, this has been reviewed. I don't want to go too much detail because today it's about plant biology. So more importantly, also nice thing about that, just want to give you actually a couple examples from this review articles where we actually can be conjugate to nuclear our DNA enzyme, either to gold particles or quantum dots, copper nanotubes, and RX nanoparticles. We can actually turn the same chemistry, either can do colorimetric sensor for lead, fluorescence sensor for adenosine, or cocaine simultaneously, all electrochemical sensors, as well as RMR contrast agents, so to actually be able to actually signaling, in this case, adenosine inside the living cells and animals. We can actually see the three-dimensional imaging of these adenosine inside this particular system in that regard. All right, so more importantly, as I said, to do spatial temporal control, we need to control the location and timing. So in this particular publication, we're able to show that we can use optogenetic controls, control not the nucleic acid, but more importantly for small molecule, in this case, zinc sensings. The way we did that is using nanoshell. They have a DNA that hybridized to the DNA so that it will not be active, because this is active from DNA enzyme. But when we add this blue strand to the hybridized to the purple part of this DNA strand to make it inactive. So we can actually deliver this one into the cells. And then even in the presence of target, in the absence of light, there won't be any kind of signal of the presence of metal ion and things of zinc. But only when you add near-R light on it, the near-R light allow its local temperature change about 5 degrees. The 5 degrees allows to this blue strand to dehybridize to the purple strand, so they actually form active DNA enzymes. In this case, only in present both near-R light and metal ion, you can see the signal, but not in the absence of either of these two elements in that regard. And also can also do without, because a lot of time when the more deeper tissues, you have to use more internal controls. This actually using enzymatic controls. In this cases, the only happen that when you have enzyme to cut this particular strand, instead of using light, we have internal expression of enzymes allow it to actually to make active DNA enzyme. In this case, only to show that you can actually see the presence of metal ion only in the presence of specific turn on of the gene expression of specific restriction enzyme in this case. And finally, a lot of time, even though we can get very low concentrations, but not for every single target. Sometimes, the targets are very, very low, we cannot even see it. So how are we going to be able to do that? We actually do single amplifications. So remember, this is an enzyme that can actually result into a product. So what we decided to do is actually the field has been booming right now in terms of DNA computations, where we can actually using this particular product of the cleavage in the present metal, in this case sodium, using as a seed to do so-called catalytic hairpin assemblies. Why want to do that? Actually, in this cases, we can using one piece of product, you can amplify hundreds of times, which can actually get fluorescence signals. You can get about 2 order magnitude increase of the signal transduction for the same product that used to be not be able to see. So now we can actually see the endogenous metal ion in the cells because it's amplification effect. And also, as everyone knows that the GIP or any kind of fluorescent protein has been very popular to be able to do signaling proteins and genes. But again, unfortunately, the application for metal ion has been very limited, maybe to calcium and zinc. But in this way, remember, we have a DNA enzyme that have been able to be specific for wide variety of other metal ions. So what we did, we took this popular the genetic encoded fluorescence system, we're able to use a DNA enzyme to be able to cleave the messenger RNA for the green fluorescent proteins. In this way, we can turn the fluorescent protein sensor, instead of sensing for proteins or nucleic acid, into the sensing of metal ion, because this DNAs are only active in the presence of something like magnesium. So we can actually now expand this very powerful methodology into a methodology for detecting metal ion and organic metabolites. So that's actually something that you can use in green fluorescent protein, in addition to detecting protein and nucleic acid, but also metal ion and metabolites. And finally, so just also another kind of recent development, we're able to actually to show that we can using photo controls to be able to actually to target the ATPs in the mitochondria, because we can actually specific using a delivery agent that only deliver our probe into mitochondria. We can using light to be able to monitor the ATP, but also in association with calcium with a reasonable spatial resolution in this regard within the cells. And now actually go to more plant biology. I think Dr. Stacy mentioned that the plant cells is much more challenging than mammalian cells, because again they have a special kind of cell walls. So therefore, to deliver these DNA into the plant cells and leaves or roots is much more challenging than developing that, delivering them into mammalian cells. So what in this particular paper, Chen Bin postdoc in the lab was able to actually take advantage of two things. One is that the plant cell has thiols in here. So what we actually find out that when you actually conjugate our DNA with the multiple thiols, that allow to interact with the surface of the plant cells to be able to make it more target delivery. But more importantly, the cell wall here also have a very narrow kind of a space. We have to choose the right size so they can actually go through these cells. So if you have distributing the disulfide unit and the right size, we're able to show that we get a dramatic increase of the delivery efficiencies, even better than some of the state of art. For example, DNA tetrahedron has been published just a few years ago to show one of the very effective way to deliver DNA into the plant cells. But our method using a repeated disulfide unit actually can even about another fold or better than that of this tetrahedron site. So in doing so, then we're able to now monitoring the glucose levels in two different kind of plant cells, and also able to show that we can actually monitor the difference of when the glucose in the regular, or the plant has a mutant of these glucose generations. We can actually monitor the differences between the two. So again, we're now actually can apply this aptamer sensor for metabolite, but also in unpublished work, we also can allow it to deliver DNA to be able to so-called gene silencing, so that actually can specifically to turn on and turn off the different genes, so simultaneously monitoring the amount of glucose level versus gene level, so you can correlate between the metabolite, like ATP, versus whatever gene of interest, and even a bit allow us to discover maybe perhaps the new genes in this regard if you're using a DNA array for these particular ones. But also this unpublished work again now can actually start seeing, instead of metal ion in the mammalian cells, but also metal ion in the plant cells. So this unpublished work, instead of using repeated disulfide linkage, we took advantage. We can actually replace phosphate diester bond with a phosphorothioate. Again, this one is enough, allowed to interact with the cell surface of thiols, allows to deliver higher efficiency than regular without regular DNA with a phosphorothioate. In doing so, we're able to monitor the zinc levels in both the plant roots. And again, we can see that, again, this is very still preliminary result, which is something we got recently, and also able to show that we can actually monitor that in the plant roots using this phosphothioate DNA, in this case, particular zinc. If we replace this nucleic DNA enzyme with a sensor for iron, for magnesium, we should be able to also monitor that. But later on, we'll also show that we can do simultaneous monitoring of these different metal ions. Why? Because we can use different fluorophores. Using green fluorophore for one, red fluorophore for another, yellow for another one, we can do monitoring of that particular ones. And finally, this is a collaboration with EMSL. I want to end with the collaboration with EMSL. So again, as I mentioned, three-dimensional imaging in high resolution is very, very essential. And so one of the kind of advancement in the nice thing about our system, we based on DNA, but our DNA is now hybridizing to another, not detecting another DNA, but instead of detecting either metal ion or metabolites. So we can take advantage of recent advancement in all the DNA detection or RNA detection field. So one really recent development was that people using so-called DNA paint technologies, and two different groups developed that. So you can actually see the high resolution here. This is 5 microns. You can imagine this is much lower than micron size. So they're able to using DNA paint technology can actually able to see, in this cases, different kind of organism in that particular regard. So this more recent work then, Chen Bing postdoc in the lab, we're able to using a DNA sign instead of red piece of DNA as a way to monitor this. So actually, we actually can do either microtubules. Again, this is his recent data. Again, you can see this is micron size. This is way below microns. But more excitingly, also this is also able to using another receptor to can actually monitor the mitochondria. So either microtubule or mitochondria. So now actually the most exciting thing that he can actually, our DNA sign, not just regular DNA. This DNA can be changing their conformations in the present metal ion like magnesium. So in this way, he actually to show that you can actually monitor the mitochondria in one color fluorophore, but also at the same time, the distribution of magnesium inside in this particular regard. He showed that you can actually see the presence of both. And but on the other hand, in the microtubule, there's not much detectable magnesium. A lot of one in mitochondria magnesium, not surprisingly, but not much in the magnesium because the color does not, no other additional color in here. But more importantly, I think Gary already also introduced this very powerful technology at EMSL. I think it's the world, if not #1, but one of a few in the US. So actually what we collaborate with De Hong and Galia to actually, they're able to show that you can using this powerful technology. So recently able to obtain a 3-dimensional image of the microtubule. But more importantly, in the next slide, we're able to show that we can see the magnesium location in 3-dimensional with a very high resolution in this particular regard. So this is, again, without the EMSL, without De Hong and Gala, we will not be able to do that. So this really allows to do the state-of-art research that really offer deeper insight into the role of magnesium inside a different one. Obviously, if you change this one into other DNA sign, we're able to actually do monitor other metal ions and also a metabolite in this regard. So that's our next step. We're interested now. We actually go into the plant cells and leaf first, and then now hopefully we can report this result in the near future. And so finally, actually I was one of the user advisory board members of right before COVID. And there's one of the report was saying that we need to know the interaction between the metabolite and other protein and nucleic acid. So I went back to the lab. We start working on this. So the key thing here is that can we actually see in the distribution not only the metal ion and a metabolite, but also at the same time nucleic acid and protein in the spatial resolution. So we can actually do so-called interactons. So the way we did that, actually, we just recently got this data that actually the binding of the target allowed to trigger the cross-linking of our nucleic acid into the proteins. In doing so, we can show that you can actually see in the absence of the metal ion or metabolite, there won't be any cross-linking, but only in the presence of the metal ion, there will be cross-linking. So we know the presence of the metal ion that next associate with the protein of interest in this particular regard. So anyway, so we just got recent result to show that the present in this case is sodium metal ion, just a proof of concept, how they associate in the cells with the different proteins. We actually did this kind of sequencing, do the proteinomic together to show that how the sodium associate. Again, we now it's just the beginning. We're hopefully using this method to be able to understand the association of the metal ion with different protein in that regard. All right, I think I'm running out of time, so I want to summarize. So I hope to convince you that Metallomics and Metabolomics are the major gap in our kind of knowledge of biology in general, particular plant biologies. We have developed a general method to be able to actually obtain DNA that will be able to recognize binding highly specifically to different metal ion or even different oxidation state of same metal ion, and also different organic metabolites. And we're able to use it for real-time imaging and monitoring these metal ion metabolites. And in addition to commercially available fluorometer, we also can use a thermometer as a way to monitor these different metal ion and metabolite in the field applications, and also develop method to overcome some of the barriers by delivering them, these nucleic acid into the cells, including plant cells, particularly using like polysulfide, and also nanomaterials allow it to actually to be able to quantify, amplify signal, and more importantly, do the spatial temporal control using either optical or internal enzymatic imaging, and also allow signal applications. And more excitingly, in collaboration with EMSL, we're able to actually show we can do super resolution, less than micron, 3-dimensional imaging of these metal ion and organic metabolites. And also recently, we're able to correlate these metal ion metabolite with the proteins, hopefully in the near future, also nucleic acid, so you can get a comprehensive picture of metallomic, metabolomic, genomic, and proteomic together, so you can get a complete picture of these species and cells, so you can actually better understanding of plant biology. Like some people did the work, particularly De Hong and Gala, and also the EMSL for the user proposals. I got a lot of advice from different people who help me help us to write proposals successfully. And again, thank you for your attention.
You want to? Okay. I want to have Heather walk way over there.
I'll start with the online question. Does the introduction of DNA enzymes into cells activate inflammations in the cells?
That's a good question. So remember, our sensors is highly sensitive. So we introduce very small amount. We talk about picomole or less amount of DNA. But more importantly, these are not, as you probably know that some of the fragment DNA can introduce these different informations. But our DNA was specifically selected to form a 3-dimensional structure, so very stable. In fact, in any of the end of our selection, we're actually using cellular extract, or in biomedical application using blood serum as a way to make sure that these nucleic acids are stable so that it will not be degraded. So they form very stable 3-dimensional structures that will not interact, specifically other than the intended target to other ones. So I cannot guarantee 100% it would not be immunogenic. But again, it's been a lot of research has shown that our DNA is much more stable, much smaller. They will not cause significant one. Another reason also I mentioned is a low amount, very small amount.
Oh, it was a fantastic collection of work.
Thank you.
I was curious when you briefly mentioned like there are more than one metal you were able to measure. I was wondering how many metals you were able to measure, and also can you comment on the affinity of your sensors? Are they able to strip the metals of any of the biomolecules like proteins or siderophores? Thank you.
Thank you very much for those questions. Very important question that we're addressing. So we have preliminary data. I can actually show you right after this. I don't have on my slide. Is we can do 4 metal ions simultaneously using organic fluorophores, because again we're limited by the overlapping of these different fluorophores. So it's a limitation. But you already know today the quantum dot won the Nobel Prize. So actually quantum dot has a much narrower emissions. So we're actually using the quantum dot as the way to do that. Then you can go to more than that. But more importantly, as you know, the DNA pen people, they can do different fluorophore time. So you collect the data of one fluorophore, you wash away, you do another fluorophore. In those way, you're not limited by just 4. You're limited only by how many washing steps you can do. So that's also another beautiful thing about our methodology. We just happen using this. We can use the same technology that developed for spatial genomic people. We can actually do the imaging for that, but we don't have data for that yet. But I'm saying that right now limited by 4, but it can be much more than that. In terms of affinity about it, that's also excellent question. I didn't have time to talk about it. We spent a lot of time. We actually designed the DNA that actually have a sensing ability with different dynamic range that are tunable. So same DNA design can be either doing the micromolar range, nanomolar, picomolar, different sequences, by the way. Why? Because when we do selections, we start with the millimolar, and then we collect the DNA, and then do micromolar, do nanomolar, picomolar until we reach the limit. And then we'll do a next generation sequencing. So we have a collection of sequences that work at different concentrations. That's very important, as you mentioned, because these metal ions, depending even location, not even cells, they have a different concentrations. So we actually have a collection of different DNA ions with a different dynamic range that change their signals. So in this way, we want to make sure our DNA ions have a similar affinity to the process that we're interested in doing. That's very important. Maybe it may not be as important for protein detection, nucleic acid detection, but for small molecule, inside the cell, outside the cell, within the mitochondria, outside mitochondria, it's completely different concentrations. So that's why we need to have a probe that customizes it so that have a concentration that's similar to the process we're interested in doing. So we actually can use two different probes that can actually allow us to do that.
All right, one more question here.
Hi, Peter Swart, Berkeley Lab. A fantastic talk. 2 questions. Maybe one of them is more related to you. The lattice light sheet microscope, does it have adaptive optics? No? Oh, okay. So you can still gain in resolution. Wow. Okay. So a related question: the DNA enzymes that you develop, can they be used in an expansion microscopy setting? To
Absolutely, yeah.
wow.
We have not done that yet, but there's no reason not to, because we do tissue samples, so we just can infuse it either before the expansion and after expansion. Yeah, I want to mention that we're still developing technology. We love to find a collaborator who can apply this one. If you're interested in any of these areas, we'd love to collaborate with you to develop technology. It's not going to work right away, as you know, in any collaborations, but there's a lot of potential here. So love to collaborate with anyone who are interested in applying technology to provide more information, more important complementary information. That's something you're interested in your system.